This is the second half of chapter 14, where we're going to do an overview of the COVID replication cycle and then talk just a little bit about vaccine development, and some other potential antiviral treatments. So the bad news is, even though this is a positive sense single-stranded RNA virus, which usually have the simplest of replication cycles, this this virus does not adhere to that normal dogma. So this one gets a little complicated. It's a big genome with lots of genes, lots of viral proteins, but I'm going to strip it down to try to make it as digestible as possible, because there's just not much point, at this level in learning what every single protein does. And in fact, they don't know. They don't have detailed functions for all of them. So we'll try to strip it down as much as we can. So we'll start just talking about the structure of the virion here. So these are enveloped icosahedrons. Maybe the defining feature of the virion is the large spike protein that you see extending from the viral envelope there. And of course, those spike proteins are going to be important for attachment, also as for fusion as well. And the spike protein initially gets translated as this precursor S0 protein, then gets cleaved into S1, S2 components. S1, we know, is the part that has the receptor binding domain that's going to interact with the ACE2 receptor on the surface of the cells. And then the S2 component is important for membrane fusion, basically uncoding, starting that whole process. So just to zoom in on the spike protein, I wanted to point out they actually have trimers of spike protein here, and the spike protein can exist in these three conformations that are each going to have distinct functions. So you have what's called the closed or down position that's shown on the left here, and they think this can essentially prevent antibodies from binding to the spike protein, right? That's obviously going to be the forward-facing protein that your immune system is going to see as virus attacks. So antibodies would most likely be effective if they were generated against that spike protein. So this is going to kind of cloak them a little bit from the immune system. Then you have what's shown in the middle here, which is the open or the up position, which you can see this little cleavage, sorry, not cleavage, but little furrow has opened up on the protein here, and that's going to allow for attachment. And then you have I was saying cleavage because then you have a cleavage event that can happen after attachment that's going to release the S1 domain and is going to give you this pretty dramatic conformational change, what's called the post-fusion structure, and that's going to allow this thing to insert into the membrane and essentially open up a pore. So that's spike protein, and then you can see within the viral envelope we have these envelope proteins shown in yellow here. These are integral membrane proteins, and although we're light on details, we think they're important for assembly of new virions in the host cell as well as the budding process. Then you have lots of this membrane protein in gray. This is the most abundant protein in the virion. It's got 3 transmembrane domains, as you can see from the little squiggle here, crosses the membrane 3 times. And again, a little bit light on details, but they think this is important for assembly of virions as well. And then the last protein to mention here is the nucleocapsid protein, the orange reddish circles, essentially the capsid protein, right? And typically we think of that as coding the viral nucleic acid, but here we have the viral RNA actually wrapped around little clusters of those nucleocapsid proteins in kind of a spiral helical format. So it is technically considered a helical virus within the icosahedron there. In terms of the genome organization, again, things get a little bit complicated. You have a large genome, 30,000 nucleotides. Remember, this is a positive sense RNA virus, which can, theoretically act as messenger RNA and be translated directly. So if we want that to happen, we need a 5' cap and 3' poly A tail, which we've got here. And it turns out part of the genome is going to get translated initially, immediately. Part of it is not. Part of it's going to be transcribed from these subgenomic RNAs that are going to be produced a little bit later in the replication cycle. But you'll notice here, from looking at the image below, you have this ORF, open reading frame, 1B, 1A. Those are going to get directly translated, right, as soon as the viral RNA is inside the host cell. Some of these others that you see over here are going to get expressed through those subgenomic RNAs that I'll come back to. So the 1A and 1B that I just mentioned, those are going to get translated as polyproteins. And as we've seen before in other viruses, then those have to be cleaved by proteases into their individual component proteins. They've found that the 1A and 1B overall give you 16 different non-structural proteins. We know 2 of those are proteases, which are going to be, again, involved with that cleavage process that I just mentioned. And then we know a bunch of them are going to form a complex called the RTC, short for replication transcription complex. That's going to, transcribe some of those other viral genes and replicate the genome, as the name implies. So just before we dive into some of these individual pieces, just to give you the kind of the big picture overview, here's our attachment, ACE2 receptor at the cell surface. Virions getting in either through fusion with the plasma membrane or via endocytosis, then fusion with the endosomal membrane. As we mentioned, right, because we have that positive sense RNA genome, we can produce those polyproteins right away. Proteases getting to work, cleaving those into their individual protein components. A bunch of those, as we said, are going to form that RTC complex. Now we're down here in the ER, which is where a large portion of the additional transcription and replication steps are going to happen. We get some membrane remodeling happening in the ER. We've seen that in a couple of other viruses, forming these what are called double membrane vesicles. This is where we're going to produce subgenomic RNAs. So we'll make, from our original positive sense, right, we're going to make a negative sense subgenomic RNA, and then a positive sense from that. And those can be used for transcription and translation of individual virus proteins. We're also going to make a full anti-genome, so a negative sense copy of the entire genome that we can use to crank out more copies of the full genome. Then, in a nutshell, you're going to have your nucleocapsid, right? Your capsid protein associated with viral RNA and that helical structure that we mentioned. That's going to basically bud into this ER-Golgi intermediate complex, which is an organelle that doesn't get talked about a whole lot, but that's where you're going to find your spike proteins and membrane proteins, etc., waiting. And then we're basically on out the cell by exocytosis. Okay, so let's talk for a minute about just the first bit of that. So we've already talked about S proteins and some of the conformational changes there, right? Binding to ACE2, etc. I told you in one of those earlier slides, the S protein gets cleaved, and they know this is happening by this TMPRSS2 protein, or depending on the cell type, cathepsin L, maybe doing that job if we're inside an endosome in particular cell types. So then you're going to get membrane fusion, right? Getting that pore produced so that we can get the RNP into the cytosol. And you can see some electron micrographs down here of that happening. So here's the virion, sitting at the plasma membrane. And we can see we've already opened up a pore here. We've already had some membrane fusion in the image on the right. And then, as you saw, we're going into the ER. We're going to do some ER remodeling and form some of these double membrane vesicles. This is an image of ER, and all these little red circles that you see are a bunch of those double membrane vesicles that have been produced in an infected cell. And again, essentially, those little vesicles are going to be replication transcription factories, right? It's just a way of kind of creating a smaller space to increase the efficiency of those processes, kind of cordon off everything that we need together. So we're going to produce negative sense subgenomic RNA, right? That's only going to be a portion of the genome, and we're going to use that to produce a number of other viral proteins that we didn't get out of our initial polyproteins. And as I said, that we're also going to make a full negative sense anti-genome copy, and that'll be the template for making more of our positive sense genomes. Then the genome's going to get exported out of those double membrane vesicles and going to associate with capsid protein and make those RNPs. And then this is showing that budding into that ER Golgi intermediate compartment, the ERGIC for short. Still a mouthful, to be honest. And you can see that happening here on an electron micrograph, right? You can see some of those spike proteins. You can see the membranes folded in, as illustrated in the cartoon down here. And you have all your RNPs kind of collecting in that invagination. Eventually it's going to bud all the way through that organelle. And, I mentioned earlier, like the M protein, we think is important for assembly here, the E protein as well, for that matter. This is also where we're going to see that precursor spike protein that we talked about earlier getting cleaved. So we're essentially maturing the spike protein here as we go along. And then we're just going to head straight into the secretory pathway, the exocytotic pathway out of the cell. So that's what was going on over here. And what's spit out, of course, is our fully mature virion that's ready to infect another cell. All right, so I stripped that down, but I think still a lot going on there. So, all right, so let's talk a little bit about vaccines. And the COVID-19 vaccine was really a wonder of modern science in terms of how quickly they were able to produce an effective vaccination. And it's worth noting, we wouldn't have been able to do it that quickly without a bunch of the earlier research that had been done on SARS-CoV-1 and MERS for that matter as well. So it had previously been shown in both of those viruses that you really needed neutralizing antibodies. Those are the, specifically antibodies that are going to attack and neutralize pathogens like viruses in order to basically stop reinfection from occurring. And they found spike protein was, not surprisingly, the only and the best protein that would give you really good protective immunity. So they had produced some earlier CoV-1 and MERS vaccines against spike protein that were both effective. So we had that as our baseline background to build on. And in the meantime, there was a bunch of advances happening in terms of creating nucleic acid vaccines, because as we talked about in our earlier vaccination chapter, usually vaccines were either live virus, inactivated virus particles, or maybe individual proteins from a virus, but there really hadn't been a lot done with nucleic acids. So, messenger RNA doesn't have to go into the nucleus in order to be expressed. So the whole idea with these nucleic acid vaccines is if you could introduce messenger RNA, you could force the host cell to translate it into a protein, then we mount the immune response against that protein. One issue is single-stranded RNA, ssRNA, can actually stimulate an immune response on its own through binding to toll-like receptors that we touched on back in the immune system chapter. So this was an issue, right? They wanted to be able to introduce messenger RNA for the spike protein, again, have it translated into that protein in the host cell, and then mount the immune response against that. So they found one way to try to force that was to modify some of the nucleosides in the messenger RNA. So from uridine to the pseudouridine to eventually this guy, N1 methyl pseudouridine. And my nucleic acid chemistry is not strong enough to know exactly why that particular transition is effective, but let's leave it at that. Seemed to do the trick. They also threw on 5' cap poly A tail, try to increase the stability. We know those structures are important for messenger RNA stability. And they tweak some of the codons as well in the messenger RNA to try to increase the efficiency with which it's being translated. So all that tweaking, bottom line, seemed to prevent this issue of immune system recognizing the nucleic acid before it was translated. So we got this vaccine, right, in rapid record speed due to some of that prior research that I already mentioned, some of these new vaccine technologies, as well as lots of funding, right? Pretty quickly, the world realized, oh, this is a serious virus. We need to address this, throw money at it. And of course, we need clinical trials, so we're lucky to have lots of volunteers willing to participate in clinical trials for these vaccines. So some of these names are going to be familiar, I'm sure. So this is pre-COVID, but 2017, Moderna was testing some of this new messenger RNA vaccine technology, and they were primarily aimed at making new flu vaccines. So they were doing some clinical trials in humans, using lipid nanoparticles to bring in the mRNA, and that seemed to be working very well. And then, of course, just a couple of years later, boom, COVID hits the scene. So 2020, they quickly switched to mRNAs against the CoV-2 spike protein. Excuse me. And right around the same time, you had another couple of giants, BioNTech and Pfizer, both putting their heads together to work on mRNA vaccines as well. So when I say this was incredible and heroic, we had mRNA vaccines that were ready for phase 1 clinical trials in humans 42 days after we had the protein sequence for the SARS-CoV-2 spike protein. That's pretty astounding. And we did do clinical trials. I tend to forget this too. I always think of the vaccine was just being pushed out really quickly. It was pushed out before FDA approval, full FDA approval, so they gave it essentially emergency use authorization before it was fully approved. But we did have clinical trials going, so placebo-controlled clinical trials, Pfizer and Moderna, with over 30,000 participants in each trial. And they were very effective. So just 2 weeks after the second dose, 95% effective in preventing COVID infections. So that's really impressive. So I mentioned FDA approval. Not to say there weren't some adverse effects, right? There's going to be adverse effects for any vaccination or drug. The common ones are what you would expect for most vaccinations. Pain at the injection site, fatigue, maybe some mild chills just due to your immune system being ramped up, etc. And then, we talked about this a little bit in class, but you do get with some very low frequency, some much more severe events like anaphylaxis, myocarditis, right? But those are thankfully rare. So overall, outside the clinical trials, just from our real world experience with these vaccinations, they are estimating the efficacy was between 75 and 95%. Sorry, got a tickle in my throat. Don't have any water on hand. And as I'm sure you know, the vast majority of hospitalizations and deaths were in unvaccinated individuals. This is 2021 data from Louisiana, Baton Rouge Medical Center. But you can see at that particular hospital, right, 91% of those hospitalized for COVID-19 were unvaccinated. 95% of those that ended up in the ICU for COVID-19 were unvaccinated. Similar number, 95% of those that ended up on ventilators were unvaccinated. So the vaccines worked, right? They were not without some risk, but going to greatly reduce your risk of serious COVID-19 complications. So then we also had, of course, Johnson & Johnson, AstraZeneca, came up with a second vaccine formula. So this was a viral vector vaccine, and basically what they were doing was using adenovirus that could not efficiently replicate in the host as the delivery vector for the CoV-2 spike protein. And this was still very effective, a little bit less effective than the other vaccine we were talking about, but still good at preventing infection and hospitalization. Again, did have some rare but serious side effects, thrombosis, right, blood clots, low platelet levels in some cases. All right, so what about antivirals? You'll see in your book there's and I'll actually show it to you in a minute, there's a big table of potential antivirals. This is still something that there's a lot of interest in. We know monoclonal antibodies could be good at reducing viral load, especially in high-risk patients, but that could depend quite a bit on which variant of COVID you're talking about. Remdesivir, which is a nucleoside analog, you may have heard of that one. It's been shown to reduce recovery time by about a third, from 15 to 10 days in hospitalized patients if you delivered it early enough in the infection, and that's since been fully FDA approved for this purpose. Then a couple of other antivirals that have shown some effectiveness, maybe lesser known. We've got a couple here that are protease inhibitors and another nucleoside analog, basically a nucleic acid replication inhibitor as well. But they, like I said, they both showed some pretty significant reduction in hospitalizations and deaths. And I don't know if we've really looked into like combinations of these things yet, but that might prove to be even more effective. So like I said, there's a lot of these drugs that people are interested in. Here's some of the ones in the top two groups here that we've talked about, at least at the time that this was put together in February 22. Remdesivir was the only one that was fully FDA approved. A handful of those others just for emergency use. And then you've got a bunch that are in, or at least were in, clinical trials, including like ivermectin, which got a lot of news coverage at the time. But bottom line is there are some antivirals that are effective and probably will be more in the future, so that's a good development. But I think that is it. Yeah.